Hi there, my name is Elizabeth McDaniel, and this presentation is all about neuroglial cells. This is for Bio 515 Human Physiology 1 with Dr. Renee Christopher at Grand Canyon University. When most people think about the nervous system, the first thing that comes to mind is neurons and neurotransmission. However, there are two main categories of neural cells, neurons and glial cells. It may come as a surprise then to learn that glial cells are actually the most numerous neural cell, making up 90% of central nervous system cells by number. This video will cover the 4 types of glial cells and the many ways neuroglial cells function to support other central nervous system cells, such as providing physical, metabolic, and functional support. And I'll also talk about how they modulate synaptic function. All right, neuroglial cells can be subdivided into 4 main categories. First, we have the stellar astrocytes, which are the most diverse functionally. There's also the oligodendrocytes, and these are highly specialized but also very important. The microglia are the immune defense cells of the central nervous system. And finally, we have the ependymal cells, which line the cavities of the CNS. And number 1 on our list is the astrocytes. These little guys do some pretty heavy lifting. The astrocytes start working from conception in humans, and they form the scaffolding for other neural cells in fetal development. In adult humans, astrocytes form the glue that form glial. That word glial means glue, and these hold the other neural tissues together. Their function is not only structural but also biochemical, and they secrete chemicals that induce the change to form the blood-brain barrier and also aid in nutrient transport to repair damaged brain tissue. Astrocytes also enhance synaptic formation and modify synapse transmission by taking up and degrading excess neurotransmitters, which halts the messenger action between those synapses. They play a central role in the glymphatic system. That's a central nervous system's version of the lymphatic system. I love the way we name things. The glymphatic system clears metabolic waste from the extracellular fluid in the brain. This is cleverly referred to as brainwashing and primarily occurs at night while we sleep. So that's one reason why it's so important to get quality sleep, so that our brain can clean itself out and get rid of these harmful toxins and plaques, which can negatively affect our cognition and our function as we age. Next up are the oligodendrocytes. That word oligo means few, and dendrocyte means a branched cell. These cells have one job. Okay, they actually have more than one job, but the most important role they play as glial cells is to manufacture those myelin sheaths that surround the axons of nervous tissue. Myelin is not only necessary for insulation of axons, it also makes a mode of neural conduction called saltatory motion possible. In the figure on the right, you can see the process of oligodendrocyte differentiation. Beginning on the left with that oligodendrocyte progenitor cell, or OPC, and growing until these little myelin-producing feet are formed. That's that far right image on the top. These myelination feet extend to neurons wrapping around the axons, kind of like little Spider-Man shoes or little tortillas around a sausage. They also have other functions. These sparsely branched glial cells include metabolic support, injury repair, and they help in our neuroplasticity. They do this because this oligodendrocyte projections, these are the legs that project those myelin feet. These are called oligodendroglia. That's a big word. And they have an abundance of a certain transporter called monocarboxylate transporter 1, or MCT1. This provides lactate to axons, so it's like providing nutrition to those axons. And when these oligodendroglia malfunction or become depleted of this MCT1 receptor, axon function is negatively impacted and neural degeneration can occur. And third on the list of glial cells are the microglia. Microglia are relatively sparse among glial cells, making up only about 10% of our brain cells. However, they play an important part of the central nervous system because they serve as the trash collectors or the garbage men, clearing out harmful debris from neuronal tissue. It's interesting to note that the microglia are derived from the same bone tissue, which gives rise to the monocytes of blood. These are the immune cells in our central nervous system. They look like wispy cells, and they have numerous long branches. These branches act as a sort of surveillance, and they monitor the well-being and the health of nearby neurons and other glial cells. And when an area of the CNS becomes infected or injured, microglia will spring into action. They roll up and move to the damaged site. Here, they are able to detect the type of infection and act either to destroy pathogens by releasing destructive chemicals or by aiding in injury repair. In some cases, they will destroy a damaged neuron completely. As immune cells, microglia remove dying neurons and they prune synapses that aren't being used. They also serve a supportive role by nurturing the survival of healthy neurons. And some research is suggesting that when microglia activity is not balanced, their destructive actions, so like if they're overzealous, can negatively affect otherwise healthy cells, and this causes excessive neuronal damage. In one study, when microglia were removed entirely from certain areas in the brains of mice, they had seizures. So those neurons just went crazy after a while. First they were okay, and then they deteriorated pretty quickly. And it's hypothesized that therapies that target microglia may be helpful to a lot of neurodegenerative diseases like Alzheimer's, Parkinson's, MS, and Huntington's. And finally, we have ependymal cells. I unfortunately don't have a pretty picture for this one because I only have the free version of BioRender. Nevertheless, these ependymal cells are epithelial-like, and that they line the internal cavities called ventricles of the central nervous system. These cells are tightly packed along these cavities, and they have cilia, which are hair-like extensions that beat, and they help propel that cerebrospinal fluid throughout the central nervous system. The ependymal cells also play a role in the formation of the blood-brain barrier, which protects the central nervous system from external bacterial and viral pathogens and harmful chemical toxins. And the most interesting thing, I think, is that these ependymal cells also function as stem cells, at least within the hippocampus, so far as we know. If you don't already know, the hippocampus is the area of the brain which is associated with memory and learning. Now, for a long time, it was believed that we only get one set of neurons in our lifetime, and once they're damaged or they die, they're gone forever. And for the most part, that is still true. Most of the neurons in our brains and in our bodies are the ones we're born with, and we have them until we die. But this was first discovered in the 1980s and then later studied more extensively. These ependymal cells, which line our ventricles, actually function not only as glial stem cells, differentiating into astrocytes, microglia, oligodendrocytes, and more ependymal cells, but they actually potentiate into full-fledged neurons. Now, unlike the neurons we're born with, these hippocampal neurons don't live until we die. They exist for a period of time, and then they deteriorate. But it's wonderful to know, especially for those of us who are aging, that we do have a sort of homeostatic mechanism to kind of rejuvenate our nervous system and our cognitive function. We're constantly producing new neurons, even if they aren't as long-lived as the ones we're born with. And no, it isn't really known yet exactly how long these neurons persist in our bodies. but some estimates are around 6 to 12 months. And here are my sources. Thank you.